Okay, good morning, everyone. So I'm really happy to be here. This was not supposed to be like this because Karim Ben-Chenan was so he is my PhD director and he was invited for this talk, but unfortunately he could not be here and he is really sorry for that, but I'm really happy to replace him for this topic. And this is really a controversial topic, like changing the mind of mice with the inception of memories during sleep. So this is a really scary title, but I hope that I will show you that it's really useful. So the work we did in Karim's lab was really helpful to understand better the link between memory and sleep. So, but just to give you a little bit more context about who I am, so maybe now I work in Cog'X, so it's a company I built with other PhD students in cognitive science to actually do some goals of that has been presented for this conference, like to try to disseminate scientific knowledge into society and also promoting awareness for the importance of some knowledge on memory and on sleep to improve well-being. So that's what we try to do at Cog'X. And for example, so what we do is that we try to give people knowledge about how memory works, so what we know now about how memory works. So for example, we did some exhibition in a company to give learners some clue about what's happening in their brain when they learn. And for example, what are the pillars, really, what they need to do to consolidate memory for a long time. So this is the 4 pillars of Stanislas Dehaene, like attention, engagement, feedback, and consolidation through repetition and spacing. So that's the kind of things we do to disseminate knowledge into society. And other things we do is, for example, this prospective report about how what are the different factors that influence sleep, and where we need to act in order to improve sleep in the global population. So this is a consulting work that we've done to try to say, okay, if we want to improve sleep in the overall population, where do we need to act? And also I still do a bit of research with Damien Léger at the Hôtel Dieu. For example, we've been working on testing the effect of circadian light in workspaces. So for example, if you have a light that is really bright but that reflects more the changing of the light changing of the sun, what effect will it have on sleep and also on vigilance at work? So that's the kind of things we do. And also we did recently, the last study we did is try to test whether some smart glasses can actually detect drop of vigilance for drivers. So this is a kind of applied research that I still do, but still I feel that I'm an outlier now today because there are lots of very intelligent researchers, and I'm a little bit. I had stepped out from academic research, but still, what I'm going to talk to you about today is really my PhD work. And so, of course, there is a link between memory and sleep, even though in my actual work it's not so easy to talk about both memory and sleep. But so quickly, of course, we know that if you learn something and you sleep just after, you're going to improve memory. And also, if you learn something but then you sleep deprived, this will impair your memory. And also, we know that when you learn something, it modifies the brain oscillations during sleep. So there is really this this very strong argument for the link between memory and sleep. And so actually the work I'm presenting is not so about Inception. That was not the goal of Karim when he designed this experiment. Actually, it was to understand better this link between memory and sleep. And the true title of this work is Explicit Memory Creation During Sleep, which demonstrates a causal role of place cells in navigation. So part of my introduction is actually what Michael just presented, so I'm going to go quickly. So first of all, there is this double dissociation that exists in humans for memory. There is this structure, which is the hippocampus, which is involved in explicit memory, so the knowledge of things and autobiographic episodic memory. And so there is the same dissociation. So hippocampus is involved in this explicit declarative memory and is not involved in non-declarative memory. And it's the same thing in rodents, where you have this huge structure, which is the hippocampus, which is involved in some explicit-like memory. And for example, one very useful model is navigation. And there is so allocentric navigation, because if you train a rat to just navigate with sequence, it doesn't need its hippocampus to do that. But if you train a rat in an allocentric memory test, it needs to know where it is in the environment to direct his behavior to find a reward or something. It needs its hippocampus. So space memory is really a useful model to study in rodents explicit, like declarative memory. And so Michael talked to you about that for the last half hour, so I'm not going to re-explain it, but if you put a rodent in an environment, within a few minutes, you're gonna, like the place cells, the neurons in the hippocampus will start firing at specific location of the environment. So in this corridor, a few minutes after the animal is exploring the corridor, as soon as it's running through the corridor, population of neurons will activate in a sequence. And so population 1, then population 2, then 3, then 4. And so this was the support of the cognitive map, really, like that the place cells really are the mental representation of space for the animal. Like that you have so strong, the correlation between the animal behavior and the activity of the cells that you can record in the animal brain is so strong that it's like there is really this cognitive map, this mental representation of space. And so this takes place during learning, during wakefulness. And so Michael talked about the reactivation that takes place during sleep and how, so this was really an approach of what's happening in the brain to make the memory consolidate. And those sequences, like those short sequence, are present during non-REM sleep, during those events, which is called ripples. And so what Michael showed, so his work, he said that when you interfere with ripples, so really this moment where those reactivations take place, you impair spatial learning. And the impairment is as strong as if you remove the hippocampus itself, so it's really, really an important role. But so this manipulation suggests that those reactivations are very important, but still ripples is really part of a global system. So if we want to address really the importance of those reactivations, maybe there is another way, which is try to see if the reactivation itself is enough to create spatial learning. So that's how Karim came with the idea that maybe we can hack the brain to consolidate a new memory. So let's say you train the animal to have a place preference. So every time the animal go to a specific place, you induce a reward. And what you see is that you can create a place preference, and you'll see that the sequence of space learning is replayed during those reports during sleep. And actually, what he decided to do is just say, okay, we're gonna let the animal navigate, and he's gonna form his mental space map. And during sleep, and only during sleep, we're going to create this new association. We're going to create the association between one particular place and a reward. And we want to see whether, if you do this specific association only during sleep, would you be able to create a new place preference? So it's like Inception, but actually this protocol was really designed to see whether the reactivation that takes place during sleep is really enough to create a new memory, a new spatial memory. So, of course, the animal is sleeping, so what we're going to use to have the place part of the association is a place cells activity. And for reward, because we cannot give any food, any rewarding reward directly to the animal which is sleeping, we directly stimulate a region which is involved in the reward, and it's known to induce a very strong very strong reward in mice. And if you do that, for example, every time the animal goes into the red circle, it has a stimulation in the medial forebrain bundle, and you see quickly that the animal just go from one red cycle to the other just to receive the reward. So it's very, very strong. And it's also doable during sleep. And so the idea was to couple the activity of one place cell, which is representative of an assembly, but for example, so the place cell here, as Michael said, so in red is this colored map is the activity of the neuron, and the red is where it's more active. And every time the animal enters the place field, so where the cell starts to fire, we have a brain-machine interface which induces directly the stimulation of the medial forebrain bundle. And what you expect is if every time the neuron fire, you associate MFB stimulation, you're going to induce a place preference. And what we wanted to know is whether we could do the same during sleep, like choose one neuron, and every time it fires during reactivations during sleep, we associate MFB stimulation and see whether when the animal wakes up, it has a new place preference. So we started by a classical wake paradigm just to see whether we affect place cell activity by this protocol. And so animals were let for 20 minutes in the environment just to form place cells and have a representation of the space. And for example, we had this cell which has a place field in the left part of the environment. And what we did after that is that we probe the exploration of the mass with 4 successive trials of 1 minute. So we put the mouse at one part of the environment and looked where it went. And same thing for those trials from different starting points. And here is the occupancy map in black and white. And you see that the animal is not preferring one particular part of the environment. It's actually exploring everything. It's not so obvious here, but in my computer it's more obvious. And so then what we did is that every time the animal entered the place field, it received MFB stimulation, like it's a coupling very short. And what we see is that the occupation change at the moment. So the animal stays in the place field in order to receive the received MFB stimulation, receive this reward stimulation. And then, in the post session, we just redo this 4 successive trials to from different starting point and see where the animal goes. And you see that now it's not receiving any stimulation at all, but it's still staying in this particular place field, in this particular part of the environment. So this is really a classical place preference. And we did that for several animals, and what you see is that the time spent in the place field after the pairing is 5 times more than before. And so now the big question is, can we do it with when the animal is sleeping with just the reactivations of place cell? So we had the exact same paradigm where we let the animal explore the environment, and it has never received any stimulation in the environment before. Then we probe the exploration by 4 successive trials, and you see that there's no particular preference in the pre-session. And then for 1 hour, every time this particular neuron will fire, every time there is an action potential, it will induce MFB stimulation, so really a reward, even though the animal is deeply sleeping. And when the animal wakes up, we put it back in the environment. Again, it never received any stimulation in this environment. Like the pot where it slept, it's somewhere else. And yet, what you see with this 4 probe trial is that it changed completely the behavior of the animal, and the animal went directly in the place field to stay. Again, it has never received any stimulation in this particular environment. So it was just the activity of the place cell coupled with stimulation that induced this behavior. So we replicated this with 5 animals, and you see that for every place field on the right, you see that in the pre-session there's not obvious preference in the navigation, and yet after 1 hour, where every spike of this particular neuron is coupled with reward stimulation, it induces a place preference for this place field, for each place field. So this is 5 different animals. So really, they never experienced stimulation in this particular environment. And what you see is that actually the result is as strong as the wake protocol. So when in the post session, in average, animals spend 5 times more in the place field than they did in the pre-session. So the new memory that we induced, the new place preference that we induced during sleep, was as strong as a normal place preference. Okay, and then we looked a little bit further at the behavior, because what we thought we induced was really a goal-directed behavior, which means the animal knows the representation of the value he wants to get and what he needs to do to get the reward. And what we know about this goal-directed behavior is that if you don't receive what you expect at the particular with your behavior, you stop doing this behavior. So that's why we wanted to see, and we looked at the trajectories of the animals. And this is one example with the first trial, where the animal is put at one part of the environment and goes directly to the place field. It's the same thing for the second trial. But you see that for the third trial, and even more for the 4th trial, the animal is not going directly to the place field. And actually in the 4th trial, it seems like it forgot to go directly. But actually it seems normal because it does not receive any stimulation anymore when it goes to it. So it seems that the memory we induced looks really like a normal memory of place preference. So, in conclusion, so we actually hacked a rodent mind, but this was really to show first that place cells still convey the same information during sleep as in wakefulness, because actually the place preference that we induced was really related to the place field of the animal when it's behaving. And this result supports the causal role of place cell activity in rodents navigation, because now you have a situation where the activity of neurons and the behavior is completely decorrelated, and still only with the activity of the place cell you can induce a place preference. So it's really a causal role. And at last, this experiment showed that it was actually possible to induce a declarative-like memory in rodents during sleep that drives a goal-directed behavior. So I would like to thank my PhD director Karim, Laurent Direg also, who directed the project, and Gaëtan, my colleagues still now. And thank you very much for your attention.